So here I have a title, Interaction between the gut microbiota and the host immune molecules. Especially my focus is the IgA secretory antibodies. So how do IgA antibodies distinguish between the good and bad bacteria for our health? Because the most researchers do not know which kind of bacteria are good or which one was a bad. But our immune system already know because through the accumulated information through the long range of co-evolution. よし、はい、これですね。はい、and then maybe you know the humans and our microbiome so should be considered as a super organism like bees and the hive. Yes, that is Rebecca's story. So because human microbiome, so for example, if we calculate the human cells, so only the 10 to the 13th, but the gut bacteria themselves only the 10 to the 14th. And then, so if we calculate the gene number, so we cannot make a so good comparison because each organism has a different gene sets, so and a different metabolic system. So that's why our body are much influenced by these coexisting the microbiome. So how to deal with these kind of big microbiome? The one is physical barrier. This is an anatomical barrier. So just the skin. So we can inhibit their invasion. The other barrier is immunity. So innate immunity and adaptive immunity. But the immune system just not only the exclude, but also the maintain the microbiome to live together. So this is very complicated system. So for example, if the immune system exposed by the harmful bacteria, so inflammation occurs. But if the immune system recognize, oh, it's a beneficial bacteria, they don't cause inflammation. So if you read immunology's textbook, so textbook says immune system can distinguish self and non-self. But bacteria non-self, among the non-self, the immune response can distinguish harmful or beneficial. So maybe to the goal of my research, I would like to know what's the criteria, the good or bad bacteria distinguishment. So then why the microbiota is very important for our health. As I see in the image, so gut bacteria produce various metabolites with their own metabolic enzymes. If you think that you intake the healthy food, but in your gut, some bacteria make the unexpected bad metabolite using their own metabolic enzymes. Then the host absorbed them from intestine. So, and then probably metabolites from harmful bacteria might cause the various diseases. So this is one example. It's published in the Nature Medicine at 2013. So especially the Western people like meat. And then, but they are worried about atherosclerosis. So, and then they think maybe red meat is okay for our health. But this paper says no, no, no. Because red meat contains a lot of L-carnitine. And then if you have a kind of bad bacteria in your gut, which produce the TMA, trimethylamine, so And then TMA is absorbed and then go into the liver, and then liver already express the enzyme to change the TMA is converted to the TMAO. And then, so it is already known as a risk factor for the atherosclerosis. So from this paper, we can know, even if you always take care of yourself with the good foods, but if you don't take care of your gut bacteria, you may cause a kind of disease. So, and then gut bacteria is usually the well-balanced community. But for example, if you take overdosage of antibiotics, this balance will be disturbed. That condition is called dysbiosis. Especially the gut dysbiosis causes various diseases, as shown here. Not only the gastrointestinal disease, but metabolic disease, cancer, diabetics, autoimmune, and the neurodegeneration disease. So, but we have the very good immune system. And among of them, so the antibody, so a kind of the IgA antibody, it's produced as a polymeric IgA, especially in the mucosal surfaces. And for example, in the gut lumen, we have plenty of gut bacteria, and then the gut tissue, and under the gut tissue, the many B cells located, and then they always make a new IgA antibodies and secreted in the gut lumen and combat the gut microbiota to regulate the symbiosis. Then 3 to 5 grams protein of IgA are secreted per day from adult gut. And this antibody is not a one-to-one specificity. They can recognize the polyreactive manner. So they have a broad specificity. So I would like to show later that one monoclonal IgA can target several different pathogenic bacteria. So they function as a blocking invasion of pathogens throughout the mucosal surface, not only the gastrointestinal tract, but the respiratory and urogenital. And then also, not only the pathogen, the IgA can regulate the commensal bacteria. That is very important to make the symbiosis, but not the dysbiosis. So, and then my research focus is I would like to develop high quality oral IgA. And then this type of IgA can suppress the harmful bacteria and reduces the unwanted stimuli caused by them, and resulting in the resetting the bodily function, because these kind of unwanted stimuli can make the chronic inflammation. This is a main cause of the variety of disease in systemically. So now we are developing this kind of recombinant IgA pentamer. And then our monoclonal IgA antibody selectively suppress the growth of harmful bacteria, but healthy bacteria spared. That's why in the limited space of gut lumen, healthy bacteria flourish and the gut homeostasis is restored. I would like to treat this kind of disease before the severe illness. So for example, so how the IgA is important for the gut homeostatic. So here there's a big cohort studies, and the IgA deficiency individual increases the prevalence of the IBD or other autoimmune diseases, like the celiac disease, the rheumatoid arthritis, and then SLE, etc. So especially Crohn's disease and ulcerative colitis, the prevalence are much higher than the IgA competent individual. So IgA doesn't do any the autoimmune, but so IgA deficiency cause the gut dysbiosis. And as I explained, probably this dysbiosis can cause a chronic inflammation. And this causes the autoimmune status like this. So IgA, the amount is very important. What is a high quality IgA? Because I already know not only the amount of IgA, but also the quality of IgA is very important to make a homeostasis. And then this may be difficult, but this is a pure immunology. So as you know, this Y-shaped protein is an antibody. And the antibody recognizes antigen with this antigen binding site at the tip of the Y hands. So, and then high quality means the antibody can recognize a proper target and then also high affinity. So, and those things are determined by this antigen binding site. So everyone has this system, and then every day in your gut, in your germinal centers, that many newly developed IgA antibody are produced. And because germline sequence can be changed by the somatic hypermutation system. So this is a system to introduce the positive mutation in the genome. Then the mutated gene can make a different shape of the Y-tip antigen binding site. That's why the binding specificity and the affinity are changed. And also, if you make IgA, you need to change this foot part, this constant region. So this replacement of C region makes the IgE and the A from IgM antibody. It's also related to the gene recombination process. So in B lymphocyte, many gene rearrangement happens upon the immune response. And then both systems essential that required the AID protein. So and then my old question was high affinity IgA and the low affinity IgA, which is important to make a good symbiosis in the gut lumen. But there's no model to answer that question, because if we knock out AID protein, so we lose IgA because of CHR also will be deficient. So, and then we screen the many randomly mutated AID protein, and I selected only one mutation. The 23rd amino acid glycine is changed to the serine, and then we made the knock-in mutant mice. So as you see here, in the fishes, the G23S knock-in mice produced plenty IgA, so class switch is okay. But if we take the B cells from the intestine and I counted, sequenced the somatic hypermutation, and it's severely down. So the G23S mice produce only low affinity IgA. And what will happen in these mice? You may know or may not this is the immunohistochemistry and of the Peyer's patch. Peyer's patch is lymph nodes on the intestine, small intestine. This is a wild-type condition, smaller than the G23S or AID knock-in mice, knockout mice. So here, AID is knocked out, and there's no IgA secretion. And then here, the red color means B cells. And then green color is a germinal center marker. So germinal center, inside the germinal center, and the somatic hypermutation is going. So the yellow cells is the germinal center B cells. And then, as you see here, G23 mice, even they produce plenty of IgA, but somehow their Peyer's patches are enlarged, and the germinal center B cell hyperplasia. So why this happens? So, and then they living in the specific pathogen-free, very clean area, no pathogen around, but they look like some inflammation. So only one thing is the gut commensal microbiota. So maybe they stimulate these mice's immune system. So then we administer these mice with antibiotics for 4 weeks. Then we calculated this number of the germinal center B cells, these yellow cells. So G23S, AID knockout mice, so these number increased, but after antibiotics treatment, it's totally normalized to the wild type level. So from this result, we can say, so probably the gut microbiota can activate the unexpectedly germinal center response. So in other words, G23S mice lost the high affinity IgA. So probably high affinity IgA is important to control the commensal bacteria. Indeed, in these mice, the gut microbiota in the dysbiotic condition. And please remember this one. I would like to compare the other one later. And then from this mouse experiment, I have to tell you one more important thing, how the intestinal IgA are powerful. So here, I compare the susceptibility to cholera toxin challenge to these mice, because wild-type mice has a very good quality of IgA, so they can survive at the 60 microgram cholera toxin challenge. But G23S mice, half of mice died at 60 microgram. It is reasonable because they do not have a high quality IgA. But, so for example, this is the lethal dosage, but if we immunize these mice with a small amount of cholera toxin 3 weeks before, so wild-type mice can survive with a twice more lethal dose. But the G23S mice happily dead. It's also reasonable. But interesting is at 60 microgram. So these mice are never been exposed to cholera toxin. And indeed, if we check in the feces where the anti-cholera toxin IgA is detected or not, no detectable anti-cholera toxin IgA. But still, the wild-type mice can survive. Why? So, because they have a lot of IgA repertoire because of somatic hypermutation. So, and some of them are cross-reactive to the cholera toxin. and that they can survive. So from this one, and I thought probably if I develop the IgA antibody, I need polyreactive and also high affinity antibody through the somatic hypermutation. So my idea is summarized here. So wild type has high affinity mutated blue IgA good antibody with a low affinity IgA antibody. Then these good antibody can trap the harmful bacteria. And then, so this is a healthy condition. In the G23S mice, they do not have a good IgA. So the bad bacteria that emerge and the overgrowth and they cause a dysbiosis. And some of bacteria invade into the host and they make a colitis spontaneously. So like inflammatory bowel disease condition. So to treat these mice, so now the clinical field, we use steroid or antibiotics or for example, anti-TNF alpha antibody. Everything target to the host immune response, just to stop the inflammation. But, so probably, I hope you already understand. It's not make a cure treatment because the cause is from the gut microbiota. So that's why I would like to treat this dysbiosis using the high quality of IgA. So just drinkable IgA antibody. Then I made many kinds of IgA hybridoma from the wild type mouse intestine. And then, so here I show the only 16 IgA clone. And here the culturable gut bacteria. And then as you see here, so this gray shows the IgA binding. So all IgA antibody can be polyreactive, not the 1-to-1 ratio. Among of these, the W27, the bound strongly to the most of bacteria, except for the Lactobacillus casei. This is a representative beneficial bacteria, Yakultobacteria. So, and this is a flow cytometry analysis. This W27 monoclonal IgA does not bind to L. casei at all, but does bind the E. coli. And if we co-culture each bacteria with the IgA antibody, only the W27 can suppress the growth of the E. coli, but spare the L. casei. So why this IgA can distinguish? Then we just did a simple Western blot. So each bacteria and the human and mouse cells lysate checked by the Western blot with a W27. As you see here, the beneficial one and the mouse cells and the human cells, W27 did not recognize almost, but here you see the clear bands. So these are all Proteobacteria species. And then actually this is the metabolic enzyme of E. coli, serine hydroxymethyltransferase, the SHMT, which is a very critical enzyme for the folate and the amino acid metabolize. And then it is well conserved between the bacteria and the rabbit. So important one. And then we distinguish which is the epitope. And then we concluded this EEHI motif is epitope. But as you see here, RQ and ELIA, SEN. So these are conserved motif from the bacteria to human. Only these 4 amino acids are different. And then Lactobacillus casei has EHNI. Only the two amino acids are difference. If we make a mutant of the SHMT, as you see here, the W27 only recognize E. coli type EEHI sequence. Then next question, which kind of bacteria has this EEHI sequence in the SHMT? And the database said the only Proteobacteria species has this sequence, they especially including many kind of pathogen, influenza, Klebsiella, Legionella, typhi, and paratyphi Shigella. So, and then these IgA antibodies are selected in vivo, and then that IgA probably can recognize all these kind of pathogen. And again, my question, why the immune system knows this should be targeted to distinguish only the harmful bacteria. So the SHMT protein expressed in the most of bacteria, but that epitope are different. So the bad bacteria only are suppressed by W27 IgA, but good bacteria spared and they can grow. So the causing a good balance of gut environment. The next, so probably this IgA can treat dysbiosis in vivo. So, and the IgA function is a whack-a-mole game. Just so some bad bacteria start to overgrow. The IgA can suppress, but do not kill. This is very important. Do not kill, and while maintaining the gut microbiota diversification, but IgA can make the balance. So here I show the in vivo experiment. I just administered the W27 IgA in the drinking water for 4 weeks. It is the same procedure as I administered antibiotics. And then counted these yellow cells, spiral patches, germinal center B cell number. And in G23S mice, GCB cells are increased, but W27 administration that normalized this. The weak antibody did not do that. But here, it's almost same as antibiotics treatment. But one big difference, because W27 do not kill the beneficial bacteria, but antibiotics kills the beneficial bacteria and then making the dysbiosis. So here, I compare each bacterial species cell number in the fishes by qPCR. And here, 2 red group is a butyrate producers, which is considered as a beneficial bacteria. So you may know the last year's Nobel Prize winner, Shimon Sakaguchi, discovered the T regulatory T cells. So this is very important immune regulator. And then, for example, in the IBD disease, inflammatory disease, we need to increase this Treg to cease inflammation. And then here, now the many researchers know that butyrate is very good inducer of Treg cells in colon. So actually W27 did not bind to this kind of butyrate producer, beneficial one. But indirectly, in the fishes, the W27 intake increased these kind of butyrate producers. And according to the increase of Treg inducers in colon in AID G23 mice and the AID knockout mice, both mice shows increased number of the Treg. So probably because of this, the inflammation is stopped. The other study also, so here is one kind of chemical can induce colitis. This is a famous chemical. But if we administer the W27, so the body weight change smaller, and then so colon lengths are maintained, and also tissues are also maintained. So because we already know DSS colitis is becoming the severer in the dysbiosis condition. So these mice are IgA deficient mice. The W27 effect is very clear. But interestingly, in the left hand, this is wild type mice. They are symbiotic condition. So W27 intake did not have any effect because W27 can bind only the harmful one. So wild type condition, that there's no target for the W27. In the same model, we can do further the colorectal polyp development. So as you see here, with the W27, here with the W27, there are many big colon polyps. But with W27, colon's number and then size are reduced. So the IgA and also the modulation of gut microbiota can cause much influence our health. So all data are mouse experiment, and then probably you may have a question. So does it work for the human gut microbiota? And then answer is yes. So for example, so IBD is related to the dysbiosis. It is well known. So here, this is a beneficial bacteria, and this black one is harmful. So healthy individual almost no harmful one, but the IBD patient has a lot of harmful bacteria. Then also their fecal microbiota diversity is reduced. So and also here, each point represent the individual. So this patient has a different set of the gut microbiota composition from the other point. This red one is a healthy control. So they are almost shrink into one group, but the IBD patients are very diverged. And then also we check these patients producing IgA can bind to their harmful bacteria or not. So, and as you see, the IBD patients IgA can bind efficiently to their gut microbiota. But we don't know. So do these IgA can bind properly harmful bacteria or not? And then, so probably I don't have time, and then I can just say the IgA index. So this is from the next generation sequencing method that we can calculate. If the IgA index is positive, that means that bacteria group is IgA binding bacteria. Okay. So for example, healthy control. So their IgA, probably good quality of IgA, can bind to the Enterobacteriaceae. This is an E. coli group. So harmful one, it's okay. And the Lactobacillus or Bifidobacteriaceae is a non-binding bacteria. That is okay. Their IgA can distinguish the bad one and good one. But IBD patient, their IgA, strongestly binding one is Lactobacillus. So somehow their immune regulation is broken. So probably in this situation, our IgA antibody can recover the gut dysbiosis. But so already these bacteria wrapped by the patient IgA. So our IgA can override or not. Then we checked. So then W27 IgA could bind the IBD patient feces like this. And then, so IgA, this IgA did not bind the healthy control, like this level. And then IBD patient, so now it's okay, W27 binding bacteria and the Enterobacteriaceae, that's okay. And the Lactobacillus is a non-binding. There also other, the collitogenic Veillonella and the Enterococcaceae are also recognized as a binding bacteria. And then, so the first part I said the IgA is polyreactive. And here I show the gram-negative or gram-positive, doesn't matter. This monoclonal IgA antibody can suppress the growth of E. coli and the Veillonella disper and the Enterococcus faecium like this. Only one monoclonal IgA can trap the many harmful bacteria. So probably this IgA, the target only harmful bacteria, and they treat the dysbiosis and indirectly probably restore the gut microbiota and suppress the inflammation. And then this is called the colonization resistance, because the W27 treatment does not break the beneficial bacterial colony. So, and if the beneficial bacteria exist in the gut microbiota, some harmful or pathogenic bacteria cannot grow well because of each interaction. And the very famous one is Clostridioides difficile infection. It is very famous in the United States. So, and the FDA already approved to treat this CDI with a live bacterial product or the fecal microbiome transplantation, because establishing the colonization resistance. So, and actually, if we check the Clostridium difficile patients, before the treatment, already, so orange bacteria emerged. This one is gram-negative harmful bacteria. So in healthy condition should be the gram-positive dominant like this. And then, so here contains many Clostridium difficile competitors exist. After the vancomycin treatment, so even their symptom is stopped. But as you see here, no gram-positive, but the gram-negative dominancy. So the antibiotics is a great discovery for human, but it breaks the homeostatic balance like this. Then, fortunately, the W27 can also target the C. difficile and then suppress their growth like this. And then we did the in vivo test. So C. difficile spores are inoculated into the mice, and then after that we treated mice with IgA or vancomycin, only for 7 days, and then stop the treatment, and then observe the other 7 days. So no treatment group died from 14 to 9. But the IgA treatment, especially W27, no mice died. And then interestingly, the vancomycin treated group, they did not show any body weight change or no diarrhea. So, but after the cessation of vancomycin, the mouse body weight reduced, and then from 9 to 5, the several mice are dead. So vancomycin, the current first-line drug for CDI, inhibits bacteria only while on medication because they can make spore and escape from the antibiotics. But after cessation, they will regrow. Then they make the severe colitis. Then here, for example, if you calculate the C. difficile, the colony number, so during the treatment, vancomycin treated group did not show any colony, but after at the last, so survived vancomycin treated mice showed emerging the CD and also toxin B production. So, and this caused a recurrence of CDI. Then the W27 treatment. So first, so we administered antibiotics. That's why it shows a dysbiotic condition, gram-negative dominance. But at last, they recover the good gut microbiota. But vancomycin group, it's not recovered because vancomycin kills most gram-positive bacteria, including C. difficile competitors. So we think not only the host health, but we think this the most closest environment, gut microbiota condition to treat many diseases, I think. So oral administration of recombinant IgA antibodies can increase the beneficial bacteria indirectly, but target only the harmful bacteria. And probably this is established, this can be established normal therapies against multi antibiotics resistant bacterial infection and also the numerous dysbiosis related disease. I would like to focus and emphasize a new concept. Now the many people in the clinical field, dysbiosis is only a condition, but I think dysbiosis is a real disease. If we can modulate dysbiosis using IgA treatment, so many people are not going into the severe real disease, I think. Probably my talk is far away the individuality, but I would say now the many researchers, especially the clinical field, and developing the new therapy is always targeting the host molecule. But real cause is not inside, mostly from outside. We need to treat outside also.